Well, welcome back, everybody. Now that we have discussed the first two parts of cellular respiration, glycolysis and the citric acid cycle, we are ready to take a look at electron transport and the generation of ATP. So let's get started. Following the first two processes of glycolysis and the citric acid cycle, NADH and FADH contain most of the energy extracted from the food. The rest has been lost as heat. These two electron carriers then are able to donate electrons to the so-called electron transport chain, and that in turn powers the synthesis of ATP, not directly, but by the creation of a hydrogen ion gradient, and then it's that hydrogen ion gradient that creates the ATP. So this sounds quite complex, and it actually is a bit roundabout. But we'll see that it's very manageable. And later on, when we look at photosynthesis, we're going to see a very similar mechanism. The electron transport chain is a series of linked proteins in the inner membrane of the mitochondrion. High energy electrons from NADH and FADH2 are donated to the chain. As electrons are passed down the chain from one protein to another, the energy in them is used to pump hydrogen ion from one side of the membrane to the other. As a result, this mechanism is often referred to as the hydrogen pump, and you'll hear that term. At the end of the chain, the electrons are passed to oxygen and thereby form water. So here's a diagram, and the way this diagram works is it shows you all the protein-like molecules. It shows you where NADH donates its electrons and FADH donates them in a different place. And it shows you the energy in the electrons. And you can see that as they go down the chain, they lose energy. And here at the end is oxygen accepting electron, binding with hydrogen ion to create water. So what's going on? Why does this happen? Well, the chain's function is to break the energy drop from the food to O2 into small steps that release energy in manageable amounts. The electron transport chain generates no ATP directly. The ATP is generated by an enzyme called ATP synthase, which is driven by the hydrogen ion gradient created by the hydrogen pump. So let's take a look at this aspect of the process, because this is still sounding confusing. The hydrogen pump is basically the mechanism that couples the energy loss from the electrons with the synthesis of ATP. So here's what happens. The electron transport chain pumps hydrogen ion from the inner compartment, the mitochondrial matrix, to the intermembrane space. The H plus then moves back across the membrane by diffusion, passing through channels in the ATP synthase enzyme. And as it passes through those channels, it provides the energy for the synthesis of ATP. Now here's a side note, and this tells you the physics of what's going on. The H plus ions have a positive electrical charge, and as a result, they repel each other. Thus, it takes energy to pump them into that intermembrane space. The energy is then stored in their mutual repulsion. You can imagine it's like cramming a bunch of elastic balls into a box. They would have energy from cramming them together. Well, the same thing, cramming a bunch of positively charged ions into a small space means they repel each other. And the energy stored in their mutual repulsion is then used to do the work of creating the ATP molecule. And now the energy will be stored in ATP. This process is known as chemiosmosis, and you don't really need to know that word. That's a word for a more advanced course. And it's basically the use of energy in the hydrogen ion gradient to drive cellular work, the creation of ATP. And you should realize that chemiosmosis is just a fancy name for facilitated diffusion used to make ATP. And make sure you remember what facilitated diffusion is, right? It's diffusion facilitated by a protein. So let's see what this looks like. Let's see what a model of this looks like. And in the next diagram, you're going to see the electron transport chain illustrated inside the inner mitochondrial membrane. And you can see the movement of the H plus ions and how they are used to drive ATP generation. So let's get situated. Here is the mitochondrial inner membrane. Here is the intermembrane space. That's the space in orange here between the inner and the outer membrane of the mitochondria. And here is the mitochondrial matrix, the yellow space deep inside the mitochondria. Now, here comes NADH and FADH, and as I said, they donate their electrons at different places. So we'll follow NADH. It donates its electrons here, and as it's passed down the series of proteins, a hydrogen ion is taken from the matrix and put into the intermembrane space. And you can see for NADH, 3 hydrogen ions are placed across the membrane. You now have a hydrogen ion gradient. What that means is you have more hydrogen ions inside the inner membrane space than here. So by diffusion, they're going to move down here. But further, all this positive charge in one place is repulsive. And so it creates a force that is going to force these things to travel down the channel with much energy. Well, that energy flowing down the channel is used to drive the synthesis of ATP by this enzyme, ATP synthase. If you really want to strictly understand the terminology, you can see that here's the electron transport chain, here is chemiosmosis, and the whole thing taken all together is called oxidative phosphorylation. We will in this course mainly use the term electron transport chain to refer to everything. But if you'd like to use the more proper and detailed terminology, feel free to do so. So here's a concept check. At the end of the Krebs cycle, most of the energy is stored in NADH and FADH2. How do these molecules convert their energy to ATP? Turn off the video, take a second to think about it, come back with the answer. So if you said they donate high energy electrons to the electron transport chain, as the electrons travel down the chain, they create a hydrogen ion gradient by pumping hydrogen ion to the intermembrane space. And then that gradient is used to drive the synthesis of ATP by ATP synthase. Then give yourself a pat on the back, because you are right. What happens to the carbon dioxide that is produced during the creation of acetyl-CoA and during the citric acid cycle? Think about that. Turn off your video. Get an answer. Okay, well, if you are a unicellular organism, it simply diffuses out of your cell. But if you are a multicellular organism that breathes, it has to travel via the bloodstream back to the lungs, where it diffuses across the membrane of the lung and then is actively breathed out. Finally, let's account for the amount of ATP produced by cellular respiration. So during cellular respiration, we've now seen the energy flow goes as follows. Glucose to NADH to the electron transport chain to the hydrogen ion gradient to ATP. All the energy that doesn't end up in ATP ends up as heat. In the next figure, we're going to see that on average, one molecule of glucose creates somewhere between 36 and 38 ATP. And that means that about 40% of the energy in glucose is transferred to ATP during cellular respiration. Here's the whole process. This is a great diagram to get used to. You can see that most of the energy becomes NADH, and then that NADH is fed into the electron transport chain, but some of it is turned into FADH2. NADH transfers more hydrogen ion across the membrane than FADH2 does, and so altogether about 36 or 38 ATP are made. 2 by glycolysis, 2 by the citric acid cycle, and then 32 to 34 by electron transport. Finally, I have 2 check questions for you. Consider the electron transport chain alone. What would the effect be if there were no oxygen getting to the inside of the mitochondria? What would the effect be on the production of ATP and on the maintenance of the proton gradient if no oxygen were there? I'll let you figure that out, but I have another check question for you. In the presence of oxygen, what would happen if you decrease the pH in the matrix of the mitochondria? That's the inner space of the mitochondria. If you decrease the pH. So first you have to figure out what it means to decrease the pH. And then you have to figure out the effect of that decrease in pH on the functioning of electron transport chain. Okay, that's it for today. I hope you now have a good, clear picture of what the electron transport chain is, how it takes the energy from NADH and FADH2 and converts it into a proton gradient, how that proton gradient is used to drive the synthesis of ATP. And of course, that ATP is critical for use in cellular chemistry that is carried out by enzymes. Well, your head is probably full on all that, so I recommend you go outside, take a look at nature, ask biological questions, bring them back to class, and I'll see you soon.